Okay, buckle up, Buttercup, because we're about to take a whirlwind tour through the history of understanding life itself. From poking around dead things to building entirely new ones, it's been a wild ride. So, grab your popcorn, or maybe a Petri dish of agar, for the ambiance. And let's dive in. Part 1, the ancient poke and prod show pre-1600s. Our story begins in a time before microscopes, before germ theory, even before hand sanitizer. Imagine a world where explaining a sneeze required invoking mischievous spirits or blaming your neighbor's bad cooking. Science was, shall we say, a bit more interpretive. Around 160 CE, the esteemed physician Galen, through his work with gladiators and animals, presumably not at the same time, decided the human body was basically a meat-powered machine full of humors. These humors, like blood, phlegm, and yellow bile, supposedly controlled everything from your personality to your bowel movements. Get a bit too much black bile, and boom, you're melancholic. Galen's theories, though wildly inaccurate, held sway for an impressive 1,400 years. That's a pretty good run for a theory that involved draining blood to cure illness. Meanwhile, over in the 4th century BCE, Aristotle was classifying living things by just looking at them. He figured out that dolphins were mammals, which is pretty impressive considering his lab equipment consisted of, well, his eyeballs. Fast forward to 1543. Andreas Vesalius, a Renaissance rock star of anatomy, decides Galen might have missed a few things. He publishes De Humani Corporis Fabrica, a groundbreaking work featuring detailed anatomical illustrations based on actual human dissections. It was like the Where's Waldo of human anatomy, except instead of Waldo, you're finding like the pancreas. This was a game changer, marking a shift towards evidence-based medicine, a concept so radical it was practically punk rock. Part 2, the Age of Reason and Unreason, 1600s to 1800s. The 17th century rolls around, and René Descartes, the philosopher who famously declared, "I think, therefore I am," decides that animals are basically just fancy clocks, complex machines, but lacking the soul and consciousness unique to humans. This was great news for anyone who enjoyed kicking dogs, less so for the dogs. Then, in 1637, Descartes publishes his Discourse on the Method, further cementing the idea of the mechanistic universe, where everything operates according to predictable laws. This was a significant step towards a scientific understanding of life, even if it did lead to some pretty questionable ethical stances on animal welfare. In 1665, Robert Hooke, using a newly invented microscope, peeks at a piece of cork and discovers tiny compartments he calls cells. This was a monumental discovery, though its significance wasn't fully appreciated at the time. Think of it like finding a crucial piece of a jigsaw puzzle, but not having the box to know what the picture is supposed to be. The late 17th and 18th centuries witnessed the rise of spontaneous generation, the belief that living things could arise from non-living matter. Recipes for creating mice involved sweaty shirts and wheat grains, because hey, why not? This theory persisted despite experiments by Francesco Redi in the 1600s, demonstrating that maggots didn't just magically appear on meat if flies couldn't get to it. Evidently, the allure of magical mice was stronger than empirical evidence. Then, in 1828, Friedrich Wöhler, a German chemist, accidentally synthesizes urea, a component of urine, from inorganic substances. This was a huge deal. It showed that organic molecules, the building blocks of life, could be created without some magical life force. It was like alchemy, but, actual science. Around 1838, Matthias Schleiden and Theodor Schwann, a botanist and a zoologist respectively, finally put 2 and 2 together, or rather cells and cells together, and formulated the cell theory. All living things are composed of cells. This was like finally finding the box to that jigsaw puzzle. Suddenly, the picture of life started to become clear. Part 3, the rise of modern biology, 1800s to 1950s. In 1850, Rudolf Virchow adds another crucial piece to the cell theory puzzle, declaring that all cells come from pre-existing cells. This finally debunked the long-held belief in spontaneous generation, though I imagine some people were disappointed they couldn't conjure mice from dirty laundry anymore. The mid-1800s also saw the arrival of the big kahuna of biology, Charles Darwin. In 1859, he publishes On the Origin of Species, introducing the world to the theory of evolution by natural selection. This theory, which explains how life changes and diversifies over time, revolutionized our understanding of the biological world and caused a lot of arguments at dinner tables. The late 1800s and early 1900s were a time of rapid advancements in biology, with scientists delving deeper into the mysteries of cells, genetics, and evolution. Microscopes got better, techniques improved, and our understanding of the intricate machinery of life blossomed. Part 4, The Molecular Revolution, 1950s present. In 1952, Stanley Miller and Harold Urey recreate the conditions of early Earth in a lab and successfully produce organic molecules from inorganic substances. This provided further evidence for the idea that life could have arisen from non-living matter, though this time it was a bit more scientific than sweaty shirts and wheat. 1953 brings us the iconic discovery of the double helix structure of DNA by Watson and Crick, with a little help from Rosalind Franklin, whose contribution was tragically under-recognized for far too long. This discovery unlocked the secrets of heredity and paved the way for the field of molecular biology. In 1967, Lynn Margulis introduces the endosymbiotic theory, which explains how complex cells evolved from simpler ones by engulfing other cells and incorporating them as organelles, like mitochondria and chloroplasts. This was like discovering that your cells are actually tiny ecosystems, which is mind-bogglingly cool. 1972 sees the development of the fluid mosaic model of the cell membrane by Seymour Singer and Garth Nicholson. This model explains how the cell membrane is a dynamic structure, not a rigid barrier, allowing for a much more nuanced understanding of how cells interact with their environment. Part 5, Synthetic Life and Beyond, 2000s present. The 21st century has brought us into the era of synthetic biology, where scientists are not just studying life, but creating it. In 2010, Craig Venter and his team create the first synthetic life form, a bacterium with a completely synthetic genome. This was like stepping into the realm of science fiction, blurring the lines between natural and artificial life. So, where are we now? We're in the midst of a biological revolution, with new discoveries and advancements happening at a dizzying pace. We're exploring the intricacies of the human brain, editing genes, engineering new organisms, and delving deeper into the origins of life itself. The future of biology is full of possibilities, and it's sure to be just as exciting and possibly just as weird as the past. So hold on tight, because the ride's not over yet. I appreciate you taking the time to explore the history of biology with me. I hope this overview provided some valuable insights and sparked your curiosity about the living world.